Yeah, I'll just do by way of introduction. So, good evening, everyone. My name's Liz Flanagan. I'm one of the senior chemotherapy sisters here at the Churchill Hospital in Oxford. And I would like to thank Elaine for joining us, and I'll do introduction in a moment, but also to the Thames Valley Cancer Alliance, who has sponsored this, the first of our 6 study evenings. We plan to run 3 this side of the summer holidays, with another 3 following. And it will be all around an introduction to the targeted therapies. So tonight we're going to begin with cancer cell biology, and Elaine, who has done her PhD in molecular biology, is very experienced at explaining the science, which I always find really good to revamp and go through, especially when we have to try and explain our drugs and how, I'm not going to say doing it even in a very scientific way, but even just in a simplistic way to explain it to our patients. But even one of the joys I like about our systemic anti-cancer therapies is if you understand the science, then you'll probably get an understanding with regards to the side effects as well. So tonight we will kick off with the first, which it'll be cancer cell biology. I've had about 40 requests asking for invites. So people are joining online or in different rooms. Kate, I hope pizzas arrived in Reading safely. Ours were late to Oxford, but I hope they're going down well. And after Elaine's done her session, I will then do a quick session on the management of an extravasation injury, which will be about 20 minutes. And we'll have questions and answers after both sessions, and then we'll wrap up before 7:30. And in a couple of days' time, we'll send round a feedback questionnaire, very simple. So please complete that in if there's any particular sessions you'd like us to do as we go forward, and then I will issue certificates of attendance. So with that in mind, keep all your mics keep on silent. We will be recording this session. If you have any objections, then dial out now and we can send you a recording afterwards. Otherwise, you've given us consent if you don't dial out. And that is it. Thank you very much. I'm going to hand over to you, Elaine. Is that okay?
Yep, that's perfect. Thank you. So for those of you who don't know me, I never quite know if people have been to any of my teaching before. As you just heard, I've done my PhD, but that was a while ago now. So I have spent a good 20 years now just doing education and training and trying to make new cancer treatments understandable to people who don't have a science background. So hopefully these sessions will be pitched at the right level. Things change all the time. So even those of you who are very experienced, hopefully there's something new that you can learn from them. This first session is on cancer cell biology. The next one is on targeted cancer treatments and the science behind them. Then there's one on immunotherapy with checkpoint inhibitors. And then into the autumn, I've got a session on innovations around antibody-based cancer treatments. You might be aware that there's lots of excitement around antibody drug conjugates at the moment. So I shall spend a while talking about those. Then session 5 is on CAR T-cell therapy for hematological cancers. And then finally, there's a session on precision cancer medicine about where we've got to, where we're going, its advantages, its limitations. So that's the 6 sessions. But this evening it is cancer cell biology. And what I've done is I've prepared a slide set. I've already sent through to Eliz the PDF of my slides so that you can have those. You don't have to worry about making notes or taking screen grabs or anything. Those slides have already been sent through as a PDF. And I promise you also, I will keep to time and make sure we don't run over. So what I'll do now is I'll just sort out my screens. I've got 2 screens so that I can have my slides on one screen and the chat function on the other screen, so that if there's any technical glitches or anything, let me know by just typing it into that chat function, and hopefully we can sort it out very quickly. So I'm just going to sort out my screens so that I've got everything on the right screen, and I shall share my screen with you if you just give me a second. And by the way, I do have a small dog. She has been bribed, but you never know. She may make an appearance, in which case the door behind me will get pushed open by a little nose and a little dog will appear. Hopefully not to speak, though. I think my husband will swoop in if she shows any signs of wanting to join in the conversation. Okay, I shall now share my screen with you. Okay, so you should have that title screen up in front of you with the title Important Concepts in Cancer Cell Biology and Genetics. These are the topics I'm going to run through in the next 45 to 50 minutes or so. I'll make sure I've got my watch on so that I can see how I'm doing for time. So I'm going to run through a lot of the basic concepts around how and why cancer happens, why it mostly happens in older people, although obviously not exclusively. I will cover how cancers evolve and diversify over the time. You may have been aware just this past few days, there's been lots of data published from the TRACERx study. I don't know if you've heard about that or not. It has hit a few news sites and radio programs, massive study into lung cancer and how it evolves and diversifies and changes over time. And Charlie Swanton, who is the king of that topic of cancer cell evolution and diversity, he's been hopefully you've been listening. So suddenly my computer just said unmute. So hopefully I haven't been muted all that time. Always difficult to see. Anyway, hopefully you have been hearing me. If anyone can quickly just type in and confirm whether you have, yeah, okay, you have. Somehow it just suddenly muted itself. Anyway, I shall continue. Anyway, so these are the topics and I shall start kind of at the beginning. I never quite know how to pitch this, of who knows what and who's learned what and when, but I find it never does any harm to go through a few basics to begin with. So we will start with the fact that cancer is caused by DNA damage. Our body is made up of billions and billions of tiny little structures, living structures that we call cells. Within each cell is an internal set of instructions. And that internal set of instructions of what to do, how to behave, when to do what, when to multiply, when to die, all those instructions are found within the cell's chromosomes. Those chromosomes are made of a long spiraling molecule known as DNA. And within that DNA are individual pieces of information called genes. So embedded within the cell's DNA, within its chromosomes, are around 20,000 or so of these genes. Each gene is a length of DNA telling the cell how to manufacture one or more proteins. And it's the proteins that do all the work of the cell. They give the cell its shape, its substance, allow it to move, tell it, force it to behave in all sorts of different ways. It's the proteins that do the stuff, and the cell knows how to make those proteins by referring back to these genes, the instructions, and those genes are embedded within the cell's DNA, which is coiled up to form structures we call chromosomes. The genes only take up about 1% of the space in the cell's DNA. So there's an awful lot of DNA and only 1% of it is the genes. So there's a lot of DNA that's not genes, and we don't honestly know what all of it does. We know what some of it does, but some of it's still a bit of a mystery. But it's the proteins that dictate what cells do and how they behave. If a cell sustains damage to its DNA, a mutation, damage, they're kind of interchangeable as words. But if a cell sustains damage to a gene, then it's now got a faulty set of instructions from which to make the protein from that gene, and it might manufacture a faulty protein from the faulty gene. Now, for some proteins, that might not matter. That might not have a substantial change in the cell's behavior, but some proteins do. For example, let's say that this is a gene for a cell surface receptor. Now the cell makes an overactive cell surface receptor, and that receptor protein sends signals into the cell telling it to grow and survive and multiply. And now that cell is part way to becoming a cancer cell because it's now behaving a bit strangely. Our cell's DNA is getting damaged every second of every day of our life. We also can be born with some damage. Sometimes we inherit damage, mutations from our parents, such as people who inherit mutations in the BRCA1 or BRCA2 genes. Sometimes as egg and sperm come together and start multiplying to create a baby, sometimes that egg and sperm have unique mutations are not found in their parent, or those mutations occur as the cells are multiplying very, very rapidly as the embryo is forming. But as well as the mutations we're born with, there's this gradual accumulation of damage in every cell as we get older. Cells are performing thousands upon thousands of chemical reactions. Those chemical reactions create damage directly, or they create byproducts that cause damage. Cell metabolism, all the byproducts of that are causing damage all the time. Cells make mistakes as they multiply. And also damage accumulates because faulty cells become less and less likely to get destroyed. And that's something I'm going to come back to in a few minutes' time. So some of the damage that accumulates in our cells is unavoidable. We could never have done anything about it. Some of it is at least potentially avoidable, although our ability to avoid it may be in our control and might not be. So exposures and lifestyle choices, hormones, infections, disease, diet, exercise, alcohol, all these things can also directly cause DNA damage, or just make it more likely that damaged cells manage to survive and multiply and don't get killed off. The upshot of all of this is that all our cells are sustaining more and more damage as we get older, and that damage is accumulating in our cells as we get older. The types of damage that happens in cells really varies. You can get all sorts of different types of DNA damage, large scale damage and small scale damage. Large scale damage includes things like when chromosomes break and rearrange themselves, the cell tries to stitch things back together and fails to do so accurately. There are also deletions and amplifications and inversions. These can affect large chunks of chromosomes, extra copies of those chunks, deleted bits, inversions where they get flipped around. You also find in cancer cells, they often have the wrong number of chromosomes. They have too many of them or too few of them. There's also a phenomenon where whole chromosomes just shatter into many pieces, and the cell tries to stitch things back together as best it can, but it can mean that a lot of mistakes happen in a very short period of time because an entire chromosome has just shattered into pieces, and nobody really knows why that happens. So mistakes, damage can be large scale, but they can also be much smaller scale. Indels, this is the name given to insertions and deletions affecting small segments of DNA under 1,000 DNA bases. But you can also have mutations affecting individual DNA bases in the DNA. These are point mutations, insertions, deletions, and substitutions. And as well as mutations, changes in the order of bases in the DNA, changes, breaks and rearrangements in chromosomes, you also find epigenetic alterations. This is when the surface of the DNA molecule has changed and that is altering the cell's ability to package up that DNA properly. There is 2 meters of DNA in every single cell in our body, and each cell is too small to be seen by the naked eye. So the packaging of DNA has to be done very tightly, very accurately, with the right bits uncoiled, the right bits wound up at any given moment of time, so that the cell can access the genes it wants and not access the genes it doesn't need at that moment in time. Epigenetic alterations, where the surface of the DNA molecule is changed, will alter which bits of the chromosome are tightly packaged, which bits are loose, and that in turn affects which genes are active and being used to manufacture proteins, which genes are being suppressed and are not being used to make proteins. Just in the next few slides, I'm going to illustrate just some of those different ways that the DNA can be damaged. This is an illustration of a chromosome translocation, where 2 chromosomes are broken. The cell tries to fix things by stitching them back together, but sometimes it gets them the wrong way around, and you can end up with a gene fusion. 2 different genes found on 2 different chromosomes now gets fused together on a single chromosome, and instead of making 2 separate proteins, the cell makes one big fusion protein. An example being the BCR-ABL fusion protein that drives the cancer cells of chronic myeloid leukemia. Gene amplification is when a section of a chromosome accidentally gets copied multiple times over, and those extra copies are inserted back into the chromosomes. You're supposed to have 2 copies of every gene because you inherit one from your mother and one from your father. But in cancer cells, you sometimes find multiple copies of a gene, and the cell uses those extra copies to make extra amounts of the protein. Classic example being the HER2 gene. In breast cancer, often there's extra copies of the HER2 gene, extra amounts of the HER2 protein, and that HER2 protein sends signals into the cells telling them to grow and multiply. Point mutations affecting a single tiny chemical, a DNA base in a chromosome, even that tiny change to a chromosome can have an enormous impact on the cell's behavior. For example, the BRAF gene contains the instructions for making the BRAF protein. A single DNA base pair changed in that BRAF gene causes the BRAF protein to be massively overactive, and you find that massively overactive BRAF protein driving things in about half of all melanoma skin cancers. So even a tiny change in a gene can have an enormous impact on the cell's behavior. Different patterns of damage have been linked to different causes, and these patterns of damage are known as mutation signatures, and scientists have discovered over 100 different mutation signatures, patterns of damage caused by different things. Some of them linked to natural aging processes, some of them linked to smoking or UV light from the sun, or the inheritance of particular faulty genes, and so on. There's all sorts of different patterns of damage that we can now link to different causes. And what we think happens is that when we're born, our cells hopefully are pristine. But as we go through life, mutations accumulate, and depending on where we go and who we are and what we do and how we behave, as well as the accumulation of natural damage as we get older, there might also be accumulation of damage caused by different external factors. And when somebody is diagnosed with cancer, and when you investigate their cancer cells, you can, if you put in a huge amount of time and effort and scientific expertise, you can tease out the different things that happen to cause those cancer cells to come into being, the different patterns of damage associated with different causes that have accumulated during that person's lifetime. So a person's cancer cells don't just contain one type of damage caused by one thing. They contain multiple different types and patterns of damage with multiple different causes, some of them not preventable, some of them potentially preventable. So cancer cells are highly complicated and tend to contain lots and lots of damaged genes. And those damaged genes are altering the cell's behavior. For example, it might be that the first piece of damage that took a cell partway along the journey to becoming a cancer cell, that might have been a fault affecting a cell surface receptor protein. That in itself, though, doesn't cause cancer. It just starts the ball rolling. But what if it was followed by another fault in another gene that means that cell is now unable to die? And what if that fault was followed by another one that means that cell is unlikely to get picked up and destroyed by the immune system? So the gradual accumulation of faults over time can lead to a cell that exhibits lots of different features that collectively cause that cell to behave as a cancer cell. Now, the features, the behaviors that cancer cells tend to exhibit, we call them the hallmarks of cancer. And they include the cell's inability to die, their ability to just keep on multiplying, the fact that they use and make energy differently from healthy cells. They can avoid the immune system. They're forced to multiply. They're able to invade and metastasize. They can create their own blood supply. So these are collectively known as the hallmarks of cancer cell behavior, and they come about because of that accumulation of mutations in individual genes. I've already alluded to this, that DNA damage tends to accumulate in our cells as we get older. And because of that, as we get older, it becomes more and more likely that a cell will have accumulated damage to its genes that's now forcing it to behave as a cancer cell. Now, that is necessary for us to develop cancer, but it's not sufficient. There needs to be a second thing that's happened. And that is, for some reason, those cancer cells are being allowed by our body to survive and to multiply. Healthy tissues, normal tissues, are very hostile towards cancer cells. The common garden cells in our tissues can sense the presence of cancer cells and try and limit their ability to grow and multiply. Also, our immune system is on the watch for cancer cells, and it's detecting faulty cells and destroying them. The reason why cancer becomes more likely as we get older is then not just about the accumulation of DNA mutations in our cells, but the fact that the hostility of our tissues towards cancer cells reduces as we get older, and it becomes less and less likely that our immune system is able to detect and destroy cancer cells that arise. So neither of these things on its own leads to cancer. You can't get cancer just because your organs and immune system are changing. You can't get cancer just because cells are accumulating mutations. But if you put these two things together, then you might develop cancer. And both of these things become more and more common as we get older. So these aging processes and impacts and all the things we're doing and the places we're going mean that mutations accumulate in our cells over time. So we might start out with all our organs and tissues being made up of cells that are relatively free of mutations. But as we go through life, some cells accumulate some mutations that give them a growth or survival advantage. And these cells start out multiplying and out competing the cells in their vicinity, such that by the time we're 70 years old or so, we are basically a patchwork of clones of faulty cells. A clone is a cluster of genetically identical cells, and your tissues become these patchworks of clones where the cells have gained mutations that are giving them an advantage over the cells around them, and they are out competing other populations of cells in their vicinity. This seems to be a fact of life. Obviously, there are things we can do that limit this as much as possible. So we can avoid smoking cigarettes, we can avoid UV light from the sun. There are also things we can do that slow down the rate of mutations and help our immune system pick up faulty cells. Things being having a healthy diet, getting loads of exercise. These things seem to help our cells remain error-free and help our immune system detect and destroy faulty cells that emerge. But you can't completely get around the fact that your immune system and the composition of your tissues does change over time. The immune system seems to gradually become less vigilant. There are fewer immune cells, such as T lymphocytes, able to destroy cancer cells, and our body becomes more and more prone to inflammation. We see this, that rheumatoid arthritis becomes more common as we get older. Other inflammatory conditions and autoimmune conditions tend to get more common as we get older. And inflammation creates an environment in which faulty cells, in which cancer cells are more likely to survive. So that increasing inflammation in our body is creating an environment in which the faulty cells can survive and flourish. So the impact of age is something we can't ever completely mitigate or avoid. DNA mutations accumulate, our immune system changes, and gradually, as we go through time, our risk of cancer increases. And added to that is this problem that cancers evolve and diversify over time. It generally takes a few different mutations that have accumulated in a single cell for that cell to become a fully fledged cancer cell. Just occasionally, you can get cancer cells driven by just one very, very powerful mutation. That is the case in chronic myeloid leukemia. That BCR-ABL fusion protein is sufficient on its own to turn a healthy cell into a cancer cell. But most of even the most powerful mutations we find in cancer cells are not on their own sufficient to turn a cell completely bad. It requires several mutations in several key genes that can cause the cell to multiply, that can cause it to avoid death, that can cause it to avoid the immune system, and so on. You need various different mutations to have accumulated to cause a cell to become a fully blown cancer cell. Some of the mutations you find in cancer cells are those that mean cancer cells are not very good at detecting when they've sustained new damage, and they're not very good at repairing any damage that arises. And that means cancer cells tend to be genomically unstable. They're liable to pick up new mutations, they're unlikely to be able to repair them, and so they evolve over time. And each individual cancer cell is evolving in its own unique way. Mutations are happening pretty much at random. They're hitting different parts of the cell's chromosomes. Just occasionally, they hit a crucial gene. They create a cell which has some kind of growth or survival advantage over its neighbors, and that cell starts out competing its neighbors and creating a new cluster of cells. And this process repeats itself time after time after time. How this happens, why it happens, the paths that cells go on, the level of diversity from one cluster of cells to the next, this is all stuff that's been elaborated on in all the different papers coming out of the TRACER-X study that have been published in the last week or so. But they're adding to our existing knowledge that we already knew that this happened, that each cluster of cancer cells is different from the next and the next. And by the time somebody is diagnosed with cancer, they have many, many different populations of cancer cells inside them, and each population has its own unique combination of mutations driving its behavior. And often, cells that have broken away and created a metastasis, they contain later mutations, newer mutations that have occurred since that cell broke away and created that metastasis. So you're going to find mutations and combinations of mutations in the individual metastases that you don't find in the initial primary tumor. Now, the reason I'm laboring this point rather a lot is that it really, really matters when it comes to trying to cure people who have cancer. Because for one thing, what you think you know about their cancer may not be correct. If you've taken a biopsy, that biopsy is only going to reflect or contain cells from some populations of cancer cells in their tumor and metastases, but probably not all. So whatever mutations you find that you believe are driving that person's cancer, you probably haven't found everything, because there are going to be pockets of cells with other mutations elsewhere in that tumor and potentially elsewhere in metastases. So a biopsy sample can't tell you the whole picture. It's one of the reasons people are excited about studying DNA that has leaked out of cancer cells into the bloodstream, circulating tumor DNA. One of the reasons people are excited about analyzing circulating tumor DNA is because perhaps you will get DNA that has leaked out of all the cancer cells in the tumor, all the different populations, and it might be more representative of all the populations of cancer cells in that person's body rather than just being representative of a subset. Obviously, as well, if you can do a blood test and find out what's going on inside a person's tumor, that is a lot less invasive than doing a biopsy. The second enormous problem we have with cancer cell evolution is that whatever treatment you give that person, whether it's a chemotherapy, whether it's a targeted therapy, whatever it is you're giving them, you are unlikely to eradicate every single population of cancer cells that exist in their body. There are going to be some populations of cells that are resistant and insensitive to your treatment because they have gained along the way mutations that mean that they just don't care about that treatment. They're insensitive to it. So whatever treatment you give, it's likely that you will leave some cancer cells behind and they will continue to grow and multiply and eventually cause the cancer to return. Now, in the next session, I'm going to talk about targeted cancer treatments. And the great thing about targeted cancer treatments is their precision. So hopefully they're going to cause fewer side effects than a more general treatment like chemotherapy. But the downside, the limitation of targeted cancer treatments is the more precise you get, the less and less likely it becomes that you're going to be able to kill off every population of cancer cells in that person's body. So chemotherapy isn't going anywhere. It's still a very reasonable treatment option. It's a more general treatment. You are killing off any cells that are multiplying. But the benefit of chemotherapy is that you have a better chance of affecting all the different populations of cancer cells. Of course, the downside is you affect healthy multiplying cells and you get a lot of side effects. But you're constantly trying to find a balance and find what is the most effective treatment strategy for that patient, because the more diversity there is in that person's tumor and any metastasis, the more quickly you're going to see resistance and disease progression. And we know that if a person is diagnosed relatively late on, when their cancer has already spread, those cancer cells are more likely to be more diverse. They're going to have more mutations inside them. And also, as time goes by, you tend to get more and more aggressive clones of cells emerging, particularly if you're treating with that person with something like chemotherapy and you're killing off all the weak and vulnerable cells, leaving behind the hardier cells that carry on multiplying. And what you find is that often treatments can kill off the more vulnerable cells and then these hardier ones remain. For example, cells with TP53 mutations tend to be ones that are difficult to kill off with chemotherapy, but with anything else as well. TP53 is the gene for making a protein known as P53. The P53 protein helps our cells sense and react to DNA damage and to die in response to DNA damage. A cell that can't make healthy P53 protein is a cell that's not going to die in response to damage to its DNA, such as that caused by chemotherapy. So if you give somebody chemotherapy, you kill off all the vulnerable cells. The cells with TP53 mutations survive, they multiply, they cause relapse. Now the person's disease is highly unlikely to respond to chemotherapy for a second time. You find this also with targeted therapies. So, for example, BRAF inhibitors given to people with melanoma skin cancer. Melanoma skin cancers tend to be caused by UV light from the sun, which causes a lot of DNA damage, which creates a lot of diversity amongst the cancer cells. So melanoma skin cancers tend to be highly diverse cancers. If they contain BRAF mutations and you prescribe a BRAF inhibitor, it will work. It will kill off all the cells that need BRAF protein to stay alive. But any cells that have additional mutations that mean they don't care about that BRAF protein anymore, they're going to survive and they're going to cause the cancer to regrow. So the evolution of cancer cells is an enormous obstacle to the successful treatment of people with cancer, particularly if they have metastatic disease, because it just becomes more and more likely that there's many different populations of cells with many different combinations of mutations inside them that cause resistance to treatment. And then the final topic I want to talk about is how and why cancer spreads. And again, this is another massive obstacle to treatment success, because as I'm sure you know, it's when people's cancer spreads that often surgery is no longer helpful, because you can't remove every pocket of cancer cells in their body, but often also systemic treatment struggles as well, mainly because of this diversity of one pocket of cells to the next. One of the problems we have in treating cancer is that cancer cells do not live on their own. A tumor is not millions and millions of identical cancer cells. There's diversity amongst the cancer cells, but there also is a lot of different cells of different cell types. So, for example, you've got whatever cells were in that tissue to begin with, common garden, fibroblasts, and other cell types that usually perform many structural functions in our tissues and probably preceded the development of the cancer. Then you've got the cancer cells. You've got whatever white blood cells you would normally find in that tissue. You've probably also got some additional white blood cell types that have been recruited in by the cancer cells. So, for example, inside tumors, you often find white blood cells that you would commonly think of as inflammatory white blood cells that are found in sites of inflammation. White blood cells like lots of macrophages, but also lots and lots of neutrophils, and maybe some mast cells and eosinophils and various different white cell types that you would normally associate with inflammation. They are attracted into tumors by the presence of cancer cells, and they actually become part of the problem. One of the reasons why treatments aren't always successful is because the inflammatory white blood cells are sending out lots of signals into their environment that are pro-growth, pro-survival, pro-multiplication signals to the cancer cells. So the cancer cells use the inflammatory white blood cells as a source of molecules telling them to stay alive and to keep growing and to keep multiplying. You also find white blood cells that actively suppress cancer-fighting immune responses. White blood cells like myeloid-derived suppressor cells and regulatory T cells. These are white blood cells that actively protect the cancer cells from the T lymphocyte. So that little white blood cell at the top is a T lymphocyte. That white blood cell has the ability to attack and destroy cancer cells, and you do find them inside people's tumors, but often they can't bring that cancer into the control. They can't kill cancer cells, but because they're being stopped by other white blood cells. Now, when I get into, I think it's the third session where I talk about immunotherapy with checkpoint inhibitors, in that session, I will be saying a lot about T cells and a lot about all these other white blood cells that you find inside tumors, because immunotherapy is our attempt to activate the T cells and generate cancer-fighting T cell responses. But this relationship between cancer cells and the white blood cells in their vicinity is massively important when it comes to response or otherwise to our standard treatments, to targeted treatments, and to cancer immunotherapy. And these white blood cells influence whether the cancer is going to spread or not. So tumors don't just contain cancer cells. They contain fibroblasts, they contain other cell types, they contain lots of white blood cells. They could also contain blood vessels, as you can see here, a blood vessel winding its way through that tumor environment. Every individual cancer cell in that tumor has its own unique combination of DNA mutations and its own unique environment, because it will be responding and reacting to signals being sent out by neighboring cells and to signals from its environment like oxygen levels and pH. And it will be reacting to the mutations it contains and reacting to its circumstances and reacting to signals sent out by its neighbors. And sometimes there is a combination of influences over a cancer cell that cause it to go through a process, a change known as the epithelial to mesenchymal transition. The epithelial to mesenchymal transition. And cancer cells that go through that process become more resilient than their neighbors, they become less attached to one another, they become more mobile. And it's those cells that are likely to be the ones that detach from their neighbors. Cells are usually making physical connections with their local environment and with the cells in their nearby environment. So they detach from their neighbors. They also start producing enzymes that digest away the protein matrix that our cells are surrounded by, called the extracellular matrix. They start digesting away the proteins in their environment, and they squeeze their way maybe into a blood vessel, maybe into a lymph vessel, maybe into a nerve bundle. They find their way into a new location, such as a blood vessel, where they will then join the flow of blood. Now, a lot of cancer cells that get into blood vessels or lymph vessels immediately die because they've now entered into a turbulent and high pressure environment. They get knocked around, they brush up against the lining of the blood vessel, and that often causes cancer cells to die. But a tiny fraction of them may survive, transit through the blood vessels, and may eventually roll along the inner surface of a blood vessel, adhere to it, and then squeeze their way out of it and start multiplying. And they will create something called a micrometastasis, a tiny new cluster of cancer cells. Now, most micrometastases never do anything. They never multiply any further. The cells in that new environment are hostile. They limit the cancer cell's growth. White blood cells might be able to restrain them. So mostly, even if these micrometastases form, they never do anything. But sometimes the cluster of cancer cells is able to recruit in a combination of white blood cells that starts providing helpful signals like the macrophages and the neutrophils. Sometimes they reach an environment that is less hostile towards them than others, maybe a protected environment like in the brain or in the bone marrow, where they're less likely to get detected and destroyed because these are protected environments. And so in this new environment, the cancer cells multiply, they create a tumor, and eventually the person is diagnosed as having a metastasis. So the things that affect metastasis are things like the genetic makeup of the individual cancer cells, the presence of cancer cells that have gone through this process that we call the epithelial to mesenchymal transition. And any cell that does that will be inherently more resilient and more mobile. The availability of nearby escape routes is going to influence whether they're likely to spread or not. And then the variety and the location and the activities of any white blood cells in their environment will also influence the cancer cells' behavior and influence whether any break away and make it to a new location. And you tend to find that each type of cancer has locations that it's more likely to spread to than others. For example, bowel cancer often spreads to the liver because the blood supply in the bowel carries blood from the bowel directly to the liver. So if any cancer cells get into the blood, the liver is where they tend to go to first. With prostate cancer, we tend to find cancer cells going into the bones in and around the pelvic area. With breast cancer, you find it spreads to nearby lymph nodes, nearby bones, but also potentially into the brain. And so on, each type of cancer has preferred locations that it tends to spread to, either because that's where the blood supply takes them, or that's where they find an environment in which they're most likely to be able to survive and multiply and cause a new tumor to form. One thing I haven't mentioned here, because it's all a bit controversial and nobody knows quite what's going on, is the idea of cancer stem cells. So there is a lot of evidence that cancer stem cells exist, but exactly what they are probably varies from cancer to cancer. How important they are and how prevalent they are probably also varies from cancer to cancer. But in most cancers, there are subsets, small proportion of cells that are inherently more resilient and more robust and less sensitive to treatment than other cells. Now, the relationship between cancer stem cells and this epithelial to mesenchymal transition causing cells to become more inherently resilient and robust and mobile, people haven't quite sorted that bit out, probably for two reasons. Probably because whatever cancer it is you study, what is a cancer stem cell is going to be slightly different from any other cancer. And also, one of the key features of a cancer stem cell is that it's inherently adaptable and changeable and alters its appearance and alters its behavior depending on its circumstances. So as soon as you remove a cancer stem cell from a tumor and try to study it, it immediately starts adapting and changing to those new circumstances, and it isn't the same as what it was when it was still in that tumor. So nobody is exactly sure how to define what a cancer stem cell is, but I think the bottom line is that in anybody's cancer, there are some cells more able to cause metastasis than other cells. Cancer cells are not all equal, and they're not all equally sensitive to treatment either. And there are some properties of these cells that cause metastasis that tend to make them less sensitive to treatment than maybe the more prevalent cells in a person's tumor. So I've tried to summarize a few of the main concepts and the sort of fundamental knowledge that helps us make sense of cancer treatments. And it's those cancer treatments that in the next sessions, I'm really going to be focusing on and building on what I've covered this evening. So hopefully what I've said builds the foundations both for understanding targeted cancer treatments and immunotherapy. I began by saying that our cells each contain an inner instruction manual, and that's their DNA, which is packaged up into these structures we call chromosomes. Within those chromosomes are the genes, the individual instructions for making proteins. Those proteins, our cells make thousands and thousands of different proteins using the information in their genes. It's the proteins that dictate cells' behavior. So if genes get damaged, if they get mutated, the cell will start making faulty proteins and it might ultimately start behaving as a cancer cell. Cancer cells are generally driven by more than one mutation. It takes multiple mutations in multiple genes in order for a cell to become a cancer cell, and those mutations have arisen through lots of different processes. Some of those processes we can control, at least to some degree, and some mutations we can avoid, but there's a lot of mutations that we cannot avoid because it is just a fact of life. Our cells make mistakes, our cells gather mutations as we go through life. When we're younger, it doesn't matter too much if cells develop mutations that cause them to become cancer cells, because our tissues are naturally hostile to cancer cells and our immune system is naturally hostile to cancer cells. But as we get older, that hostility and that ability to detect and destroy cancer cells lessens over time and mutations are accumulating. So cancer is mostly a disease of older age because of the accumulation of mutations, because of the way our tissues change, because of the way our immune system changes. And we have a couple of features of cancer that make it a really, really difficult disease to cure. One is that cancer cells tend to evolve and diversify over time. So a person's cancer cells are actually many different populations of cancer cells with their own combination of mutations, their own unique vulnerability to treatment. So there's lots of diversity within the cancer cells or between the cancer cells in a person's body. And also they create for themselves an environment that suits them, an environment that nurtures their growth, an environment that protects them from cancer killing cells of the immune system. So you have these two enormous obstacles to successful treatment: the diversity amongst cancer cells and the way they create an environment that supports and protects them. And if cancer cells have metastasized and spread to new locations, they become even more difficult to treat. Each metastasis is likely to contain a unique combination of mutations. It's going to have its own unique environment. And it tends to be the more aggressive and more resilient cells that cause metastases. Okay, I'm going to stop there because I wanted to allow plenty of time for any questions that you might have. And I also presumed that nobody was going to argue with me if I kept to time. So I will stop sharing my slides there.
Thank you, Elaine. That was great. I actually, I really just enjoyed. You sort of just think of cancer cells as developing, mutating, but it's putting it into context as well with our immunity. As we get older, our immunity reducing, and that was just a brilliant recap on how cancer cells develop and how. And I loved about the spread of cancer and like the little blood channel and them all, and never realizing the importance of inflammatory white blood cells. So thank you very much for that. Does anyone have any questions, please?
You're very welcome to unmute yourselves or use the chat function. I've got that open on my screen if you've got anything you'd like to clarify or ask or want to comment on. You're very welcome to do so. I think that emphasis on the immune system has really changed over the years. I mean, I'm deep into writing the second edition of my book, and I've even changed the title of it. So it's no longer a beginner's guide to targeted cancer treatments, but it's a beginner's guide to targeted cancer treatments and cancer immunotherapy, because I think the emphasis has changed. And something that really struck me the other day when I was reading was that the evidence that even if somebody has surgery or chemotherapy or radiotherapy, whatever treatment they have, if they're cured of cancer, their immune system will have gotten involved somewhere along the line. You will never, through your treatment, have got rid of every last cancer cell. It's just highly unlikely that you've gotten everything with your treatment. And so really, if somebody is cured, then probably their immune system took part in that whatever type of treatment they had.
And what your session is also highlight is the importance or the role that combined chemotherapy and immunotherapy or targeted therapy play together, which is really being clarified with that teaching.
Yeah, I think that's another really fascinating topic. I go around in circles. I must admit, I sit and spend a lot of time staring at clinical trial results with combination chemotherapy and immunotherapy. And in my brain, I flip-flop backwards and forwards. Is it a good idea? Is it a bad idea? What do these results say? Are people better off? Are people not better off? And I do go round and round in circles because there's so much logic to it. You know, the chemotherapy kills the cancer cells. It creates lots of debris. That debris helps activate the white blood cells. Also, chemotherapy damages the immune suppressing white blood cells, the inflammatory white blood cells. Like, there's lots of very logical reasons why that combination is a really good idea. And I think for some people and in some circumstances, it is a really good idea. But you can't get away from the fact that chemotherapy, a lot of chemotherapies damage your immune system. And so they're not entirely synergistic. They're not always pulling in the same directions. And sometimes you see lots of benefit in short-term survival and in the proportion of patients who benefit, and you see lots of good stuff happening when you combine the two. But then you look at the data a few years later and you think, oh, was that definitely the right idea? And I think particularly in something like lung cancer, where you've got a proportion of patients with very high PD-L1 and very high chance of benefiting from immunotherapy, you do want to give that alone and not include in the chemotherapy as well, because you've got the best chance of the immune system really controlling things of its own accord without any impact of the chemotherapy. But some of these things, you know, it's still early days for immunotherapy. A lot of the clinical trials we've got data from, you've still got maybe 3 years of follow-up or 4 years of follow-up. It's only a handful of clinical trials, like some of the melanoma trials, where you've now got 10 years of follow-up. And you really start to see how things panned out over that longer period of time. So I think there's an awful lot we've still got to learn with some of these combinations.
And I think and even with any of the targeted therapies, but yes, we're in early stages yet. And just understanding how the cell biology there, just actually how much we've still got to learn about it all.
Oh, I can just see somebody.
Oh yeah, it's Mark Full. Yeah.
Just wondered what you thought about younger patients given immunotherapy earlier in their lives. Do you really understand the effect of this? Yeah, and I think one of the things you have to bear in mind in immunotherapy, particularly the checkpoint inhibitors, is that you are boosting T cells. Now, sometimes your body has a really good reason for wanting to suppress some T cells because they would cause autoimmune disease, because they would cause chronic inflammation. So your body is at any point in time suppressing some T cells for your benefit. And with your checkpoint inhibitor, you are boosting T cells wherever they are in the body and suppressed for whatever reason. And so, you know, you can end up with T cells becoming active that destroy your thyroid gland or destroy your pituitary gland, and that gland is never coming back. And so, I mean, I've been just grappling today with the section of my book about biomarkers for checkpoint inhibitors, and there's no perfect biomarker. We have no perfect way of predicting who is going to benefit from immunotherapy and with checkpoint inhibitors and who is not. And ideally, you'd want to avoid giving it to people who it doesn't benefit. At the moment, that's the majority of the people we give it to. So I think it does have to be part of the equation for younger patients, is that you can leave them with lifelong problems. And at the moment, we're not very good at selecting or predicting who should receive them and who shouldn't. So I think, you know, I suppose the younger patients given immunotherapy at this present point in time, it's mostly younger people with melanoma skin cancer given checkpoint inhibitors. And then realistically, immunotherapy, if they've got advanced disease, it's the only thing that can cure them. And therefore, I think the equation is relatively straightforward. Unless, Mark, you've got another scenario in mind. I don't know if you were thinking just about checkpoint inhibitors or other forms of immunotherapy. But I think their long-term side effects do have to be taken into the equation.
Mike, he doesn't have a mic, so
Yes.
Mark can't.
He might give him add any
give him a
clarification.
couple of minutes. He'll type a reply in. We've got
I
another.
just see another couple of questions there.
Yeah. Are there any perspectives of mimic the ability of cancer cells to resist, survive, or avoid death in order to produce super cells to be used, for example, during bone marrow transplantation?
No, I mean, I think the danger is that as soon as you start mimicking any of those properties, you run the risk of creating new cancer cells, because those really resilient cells are dangerous. I mean, I think with bone marrow transplantation, there are efforts to modify the cells that you put back into the body, or modify the cells you put into the body so that they're less sensitive to some treatments, so that you can use very, very powerful treatments for bone marrow ablation prior to receiving transplant. And you can afford to go really hard because the transplanted cells are resistant to your treatment. So I think there's the odd thing you can do to sort of mimic, I guess, some of those features, but you've got to be really careful, because, you know, there have been, I mean, one of the sessions I'm going to come to in the autumn is about CAR T cell therapy, and just occasionally a person's cancer cell sneaks into CAR T cell therapy and you end up causing a massively aggressive leukemia that kills that patient. So you've got to be really careful. So just seeing a comment from Nikki.
Yeah.
Adjuvant chemo or not? Yeah, I think that is a really difficult choice. I was just talking to somebody about that the other day. And I think, you know, if it's a younger person and their cancer cells are really aggressive, then sometimes you're going to have to hit them as hard as you can. But there are some times where adjuvant chemotherapy, it's really not adding that much, and you know, you need to have that really difficult conversation with somebody, especially if they're, you know, in their 80s or something, like exactly what are you achieving with that adjuvant chemotherapy. I think those choices are really difficult because there's no perfect answer there. I mean, things we can do now in breast cancer are giving more accurate predictions of relapse, but you don't want to overtreat unnecessarily. And if it's a marginal difference, then for some people it won't be worth it. And I think they are difficult conversations because, you know, it's all statistics, isn't it? It's all numbers, and that can quickly just bamboozle people. And even your voice and your body language, the way you put forward the information, speaks volumes to a person who's not actually understanding the numbers that you're giving them. So they're really difficult conversations to have. Oh yeah, neoadjuvant pembrolizumab in breast cancer. Yeah, I mean, there is, I think, increasing evidence about neoadjuvant checkpoint inhibitors, that is a really good time to give a checkpoint inhibitor. If you're going to give one, then to give it at the point in time where the tumor is intact, the lymph nodes are intact, the lymph vessels are intact, the immune system is intact, there's been no damage done by any treatment that you've already given. This is the time when you're going to activate the most T cells. You know, by the time you're giving adjuvant checkpoint inhibitor immunotherapy, you've just removed the tumor and possibly the lymph nodes and the associated lymph vessels. So exactly what T cells are you hoping to activate when you've just removed all the T cells inside and in the vicinity of that tumor? So with adjuvant immunotherapy with checkpoint inhibitors, you get far fewer T cells activated than neoadjuvant treatment. So we're seeing, for example, in lung cancer, that neoadjuvant checkpoint inhibitor therapy has long-lasting benefits. We've seen in melanoma skin cancer that neoadjuvant immunotherapy is better than adjuvant immunotherapy for preventing relapse. So I think neoadjuvant treatment, I mean, you have to have the evidence because you don't want to be treating people unnecessarily. As I've already said, checkpoint inhibitors can cause lifelong side effects. You do not give them lightly. But if there was ever a time when they were going to have an impact, it is neoadjuvant. So I think we need to see more trials over longer periods of time to be sure that we're doing the right thing with it. But I think it's more exciting as an option than adjuvant checkpoint inhibitor. But I think it's still early days with that.
And for all of us who work in day units, we take a deep breath in at the realization of more neoadjuvant work potentially.
Yes, from what I understand, it adds a lot to your workload.
It just makes it different. Yeah. I don't think there's any more questions come in. So to keep us on target, thanks very much, Elaine. That was great. And we've got you back on Thursday, the 27th of April, which is in about 10 days' time.
Yeah.
And that session is, sorry, I've just moved my
What
piece
about
of
targeted therapies? So
targeted therapies.
Matching mutations with treatments and
that's it. Yes. We look forward to seeing you then. Thank you very much.
Perfect.
And for the rest who would like to listen to extravasation, I'll do a quick 15-20 minute update on it now. Please stay on the line. Thank you.
Okay, bye for now.
Thanks, Elaine. Bye.
Bye.
Hi, everyone. So, for those of you who don't know me, I'm Liz Flanagan. I work here in the OUH, and I'm going to do a quick 15-minute session, 20-minute session on the management of extravasation. Now, I'm not going to cover prevention of extravasation in this session because that in itself, whilst I feel is extremely important and as important as the management, I just don't have enough time, and I think we all want to get home. But at a later date, if anybody ever wanted to go over prevention, I'd be more than happy to do so again. So during this session, we'll just go back over the definition, how to identify treatment, go through a case history, and just a little photo warning. Some of the photographs, admittedly, whilst some I've got off Google, others are actually from scenarios of extravasations that over 35 years of giving chemotherapy, I have actually seen from time to time. So what is extravasation? It is the inadvertent leakage of a drug into an area where it shouldn't be. So this is where if it's an IV injection we're giving, most commonly it leaks into the surrounding subcutaneous tissues. And we may also refer to it as tissuing or infiltration. And when we use those two terms, we tend to think about our normal saline drugs, our non-anti-cancer therapies. And when we talk about extravasation, we tend to be thinking about our anti-cancer therapies. It can be acute, delayed, and distant. Acute, very obvious, we see it there and then as it's occurring. Delayed is where the patient presents a day, 3 days, a week, 3 weeks later with symptoms and signs of an extravasation injury. And distant is where you may find it is delayed, or that if we cannulate, doing a cannulation and start to administer the drug below a previous attempt, that you can then see the injury happening there and then. But the important thing to remember with extravasation injuries, that a lot will heal by themselves, very much dependent on the volume that's been extravasated, the concentration and the type of drug. And it's a small proportion go on to cause damage. However, it's in a small number of cases they can go on to cause a serious amount of damage that can last for weeks, months, and be permanent, and can cause damage to the nerves, local tendons, and to the joints. But in this picture, you'll see some shedding of skin, and that can be quite common, dependent on the nature of the drug and the volume infused, is that dry, scaly skin and shedding. And in the worst case scenarios, we may need to consider surgical intervention, which could be a surgical washout. We may look at surgical debridement and skin grafting, and worst case scenarios, even amputation. Now, I've never, touch wood, had anybody requiring an amputation, but I have seen one patient over the years who've required skin grafting. The incidence is not, it varies, the literature varies, 0.1 to 7%, but there is no, for us here in the UK, there is no national register, there's no way of collecting nationally the statistics on extravasation injuries. So it's a bit of an unknown entity. But I think what we've got to bear in mind is, let's look at what's going on in our day units on a day by day. How many different drugs are we giving? How many different infusion bags? How many different bolus injections are we giving? And what is the rate of extravasation in those cases? And then, is it that we've suspected an extravasation, or is it an actual extravasation? So one of the big things that reduced, because I do remember from 30 years ago, when you'd be walking around, you would see people with extravasation injuries. And even for those of us who've been nursing for a while, can we remember all the times with just even saline and glucose infusions, antibiotics, how many patients had tissuing, infiltration, and developed a redness and a swelling that lasted several days. And that was even just with our non-cytotoxic drugs. So that's all been reduced due to training in the prevention, and also the monitoring, doing our VIP scores. All of that have contributed, and the big contribution to reducing extravasation injury has been in minimizing the number of people who can actually administer the drugs, which in consequence is mainly nurses who have actually been trained to administer and watch out and prevent and use techniques or assessments of risk and assessment skills to actually prevent extravasation injury. So the classification of drugs varies. What we can have is some of the literature. We will have what we call our vesicant drugs. So these are the drugs here on the left-hand side in red, and there's a list of them, and they can be what we call the DNA binding drugs, or we have the non-DNA binding drugs. The next group you'll have are what we call our irritant drugs, and we can have our non-vesicant drugs. So into our non-vesicant drugs, which is on the right and in green, we will have a lot of our monoclonal antibodies, our immunotherapies. We will have drugs like cyclophosphamide. Going to the vesicants, the DNA binding drugs, they are the severe category, and they can cause a huge amount. They would be the worst ones, in my experience, for the damage that they can cause. And really, we do need to think about, when we're administering these drugs, which would be covered all in the prevention, is about the volume, how we're administering them, where we're administering them to, and actually to prevent that. The vinca alkaloids, the non-DNA binding ones, they do cause blistering and ulceration, but patients tend to make a recovery from that. In the irritant ones, I feel the ones to be mindful of is the ones divided into 3 categories here. And over on the left-hand side of the amber section is just look at the list of drugs there. We have drugs like oxaliplatin, mitoxantrone, not used so much nowadays, but cisplatin, dacarbazine. They are our infusional drugs. And in experience, what has shown is that when these extravasates, they tend to be a bigger volume of fluid as opposed to some of the other drugs that we give. So it's just being mindful, whilst they're not vesicant, if you get a big extravasation injury with something like oxaliplatin, which is what I've seen, the damage can be severe and limiting mobility for the patient and take several months to recover from as well. So here we just have a brief explanation of why our drugs are vesicant. So we have our ability to bind to DNA, which is our anthracyclines, the ability to kill replicating cells. Do they cause tissue vascular dilation, constriction? What's the pH? And is the osmolarity greater than that of plasma, so that you get that leakage of the drug out into the veins? These are all factors that will contribute to the extent of injury that these drugs can cause. It's also being mindful as well, which is what I've already said, is that we have non-cytotoxic drugs such as our electrolytes, mannitol, which we give routinely pre-cisplatin, Venofer, our antibiotics, if they're in high concentrations, can also be of a vesicant nature as well. So the signs and symptoms to look out for: stinging, burning, pain, swelling, oedema, any acute change at the site of the injection of the cannula. And never underestimate this. And we all have experience of the patient who were injecting a drug and they start to make facial changes. I always think, just quit, with their eyes, you'll see the sudden facial changes and that they start to go, it's sore. Always act upon that and investigate. Is there any redness at the site? Has the pump, or even if you've been injecting it as a bolus, what are the pressures on the pump? Is the pump constantly alarming? Where is the location of the cannula? What movement is the patient doing? These are the symptoms, but it's really the stinging, burning pain. Think about when you've all had flu, COVID vaccinations. They all sting initially. So if it ain't going into the vein, ask our patients, what do they feel when we've just cannulated and you've injected some saline to flush the cannula? They all say they feel some cold running up their arm. But if they start to complain of burning or stinging, then look out, check for extravasation injury, and if in doubt, don't proceed with your anti-cancer therapies, especially if they're vesicant or the exfoliant drugs. Seek further advice, support from colleagues. One of the things is sometimes you may suspect an extravasation injury and we get blood coming back into the IV line. And it's not always the presence of blood doesn't mean we haven't got an extravasation. Be mindful of that. It doesn't happen most times. if it has extravasated, you don't get blood flowing back into the IV line, or when you try to aspirate, coming back into the syringe. But the presence of blood is not, it can be a sign, can still be a sign of an extravasation injury. And then we talk about blistering, but blistering will only come on about 2 to 3 days after the injury event. In particular with anthracyclines, it's more common for patients to present 2-3 days later. If you look at patients with docetaxel who can get chemical phlebitis and get patches of redness going up their arm from where the cannula was sited, that will take several days to a couple of weeks to appear. So when is it not an extravasation injury? Well, the times we've got to think about is anthracycline flare. And this is when we're giving our anthracyclines through a peripheral cannula. You might get a mottled red rash going up along the vein, and it's irregular in shape. And where any of the veins truncate off, it'll tend to be white over there, those areas, and generally responds and stops spreading once you stop infusing the anthracycline and infuse with some saline, and it gradually disappear. Once we've seen it with cisplatin, which is the bottom picture here, and you can see this whole arm became red and mottled, and in that case we stopped. We gave the patient hydrocortisone and Piriton, and when they recovered after about half an hour, we then resumed the cisplatin infusion at a slower rate. Another problem that patients can complain of is vein pain. And with dacarbazine and gemcitabine in particular, those of you who've been administering either of these drugs will have seen this and heard patients complain of it. And oxaliplatin, because it can cause a peripheral neuropathy, a cold neuropathy, they too can complain of cold around the cannula site. Dacarbazine and gemcitabine, when we stop the infusion, the pain tends to resolve, and you can restart the infusion at a slower rate with a warm pack, and in some cases even hang another bag of saline and to piggyback onto the infusion, and that will help to prevent recurrence of the pain. And I liken, when asking patients to describe that pain, it can feel like that really clawing, clamping pain that you get maybe when you've eaten ice cream too fast and you get a pain here in your forehead. For patients with dacarbazine and gemcitabine, they'll describe it as that cold clamping pain. Oxaliplatin is different because it causes the drug itself causes this cold neuropathy. So stopping the infusion, symptoms don't tend to resolve so quickly. You can piggyback some glucose, and you can apply a heat pad, and that can help as well to prevent this cold. But more and more units are moving towards use of PICC lines to administer oxaliplatin. So the signs and symptoms, as I've said, is the patient complains of burning, stinging. You might have actually noticed some redness, the shape of an egg. It tends to be, if you see in this diagram here, it tends to be a regular bump. It can be quite acute when you see it, and you definitely know it's leaked into the surrounding tissues. So what are we going to do? Well, once we've arrested our own breath and thought and got over our initial panic, it's actually make sure we stop the infusion, assess the situation. And at the same time as doing this, we're trying to reassure the patient, but request assistance from colleagues. Ask them to get your extravasation kit that every unit will have in their department, and try to be thinking, well, assess what is the drug that's infusing, where is the location, is it on the back of the hand, is it in the forearm, is it what volume of infusion are you looking at? When your extravasation box or kit arrives, then using a 10 ml syringe, try to aspirate back some fluid. Disconnect, or if your cannula is such that you've got another bung, another arm to your cannula, try to aspirate back 3 to 5 mils of fluid. In reality, if it's a full-blown extravasation, all you're going to do is get bubbles coming back. Sometimes you might get some blood flow if the cannula is still in the vein. Put a ring around the area that is swollen, just so that you can begin to watch the extent of the swelling, but also the redness. Dependent on the nature of the drug and dependent on what your own local policies say, remove the cannula. So for drugs that are neutral or irritant, you can remove the cannula. And at that point, start to elevate the limb. And then think about applying either a hot pack or a cold pack. If you can't remember which ones to apply the hot pack to, the two really to think about is the vinca alkaloids and oxaliplatin. If you were to inadvertently put a cold pack on a vinca alkaloid extravasation injury, it's not that it will cause any harm. Certainly you wouldn't put it on somebody's arm with oxaliplatin, as the cold, they won't tolerate it. But the whole idea really of a hot pack is to disperse the drug, and cold packs is to localize the drug to that area and localize and keep it all in the one area. Fundamentally, the evidence behind hot packs and cold packs is vague, but on the other hand, there's nothing to out there in the literature that says don't use it. And if nothing else, it sort of it will provide some sort of comfort or pain relief to patients as well. For all extravasations, have your doctors come and assess. What you want to be thinking about is what's the volume that's extravasated? Where's the site? So what's the vesicant nature of the drug? And then they'll prescribe drugs and they'll be administered according to local policy. And I'll go through some of them in a moment. For vesicant drugs, think about and even large volume exfoliants, irritants, thinking oxaliplatin in particular. Contact the plastic surgical team, inform your consultant, and at this point you will be filling in your patient information, give the patient information leaflet, arrange follow-up, and at a minimum follow-up should be 24 hours and 7 days later. And our documentation, plus or minus photographs and complete incident form, and it should include all of this information that I've just listed above. When it comes to antidotes, dexrazoxone, or sometimes known as Savine, is the only licensed antidote for an anthracycline extravasation. It is a cytotoxic in its own self, and it needs to be prepared in an aseptic surfaces. Now, some areas may have closed systems that allow the transfer and the compounding of the drug itself. Here within the Thames Cancer Valley, we haven't done that. So the drug needs to be prepared in ASU. The patient will require a consent, and ideally it should be administered within 6 hours on days 1, 2, and 3. So you cannulate the patient in the unaffected arm, and it is given as an hour infusion, and it's 1,000 milligrams day 1, 2, and 500 milligrams per meter squared on day 3. The thing to note is it arrives in a glass bottle. So it's just being mindful. The only time, well, we've had it used twice here in the OUH, is that it the first time it was delivered, it was dropped by the pharmacist who was delivering it to us, and the glass bottle broke. But we had several mishaps with that day 1, 2, 3, that particular time, whereas on another occasion it all went smoothly. But just remember, it's the glass bottle and the packaging it arrives with to hang it on the drip stand may be not the most stable, and you may need to be creative with how and do a bit of DIY to make sure it's securely attached to the drip stand. And the typical side effects of it are as per cytotoxic drugs, but you give make sure your patient has some antiemetics, and it can cause neutropenia. The other drug that is licensed and used in particular for vinca alkaloids is hyaluronidase. It breaks down the collagen in between in the cells, and therefore makes the tissue in the area of the extravasation. By breaking down the collagen, it makes it easier for the drug to be absorbed and dispersed. Ideally, we're looking at that being given within 1 hour of the injury, and dependent on the extent of the injury, you'd give 1 to 6 mils, about 150 units per mil with water for injection, and you might do that over 2, 3, 4, 6 injections, always remembering to change the needle every time you reinsert the needle. So it can involve the patient having several injections, but any time I've seen it being used, it has prevented the ulceration of a vinca alkaloid extravasation injury. In the past, some of you may have heard, and I keep this in here because sometimes you may come across people and go, well, why, what about dexamethasone subcut or hydrocortisone cream? And the evidence with the last EON's ESMO guidance on extravasation injuries, the evidence wasn't concrete. So since then, they've been removed from the guidance. And in some trusts, you may see DSMO 99% being used for anthracycline or certain other vesicant drug, and that's where you apply this liquid to the injury, and it freezes the cells in that area and stops the molecules from spreading to surrounding tissues. But the reality is we're supposed to use DSMO 99%, and in the UK only 50% is available, and therefore it's not included in the TVCA guidance. But I just say that because you will see it written in other people's policies. Other treatments and aftercare. So for vesicant injuries, if you haven't or you're not able to use Savine or hyaluronidase, refer the patients to the plastic surgeons, and they will then consider a washout technique. I don't know if any of you have ever seen one or how familiar you are, but just looking at the picture here on the right, you'll see the several little puncture holes, and then they squirt volumes of saline, and really it's what it says, you're washing the drug out. But of course, what you've got to be mindful of here is consider the PPE that everybody is wearing, certainly to be wearing goggles and full theatre gown and be well protected, because there is molecules of cytotoxic being squirted out from here, and keeping gauze squares, CADs, or Inco pads in the area, and reminding, I have seen a surgeon who launched in one time to do a washout and didn't, apart from an apron, wasn't really thinking about anything else, and we had to stop him from going any further till we've had the appropriate PPE put in place. As I've said, cold and heat packs. Make sure they've got a covering on them to prevent either burning the patient or causing frostbite, and apply 10 to 15 minutes, 3 to 4 times a day for 24 hours or until recovered. One of the things from the plastic surgeons is moisture the skin, and just keep applying, and certainly with any extensive injury is keep applying plenty of moisturizer at least twice a day, if not 3-4 times a day. And then as any redness and swelling begins to subside, to think about massaging it into the area to stop the skin from fibrosing and becoming hard. For any extensive swelling, also elevate the limb, keep it up high. Think about pain relief for your patients. Ibuprofen gel is okay, and even the use of paracetamol. But depending once again on the extent of the injury, the nature, appropriate medication will pain relief will need to be thought about. And your follow-up is really what we do here is we try and follow the patients up the day after, well, not try, we do, follow the patients up the day after their extravasation injury. And we will continue to telephone them and do a telemed assessment every 2-3 days until we feel the injury has started to recover and until they've returned to clinic to their next outpatient appointment. And when we're assessing them, we're actually looking at the pain, the level of pain they've got, the range of movement. Is there blanching in their fingers? How's their hand movement? And we will keep that follow-up going until such time as the patient has fully recovered from that injury. So this is a gentleman who suffered a docetaxel injury, and you can see this extensive bit here. So docetaxel is an irritant drug. He was a gentleman who rested his arm on the armrest of the chair and didn't notice it swelling, didn't notice anything. In fact, he did notice, but then he didn't want to bother the nurses, and he and his wife then had a fallout over it. But at the point we came to it, he had an extensive swelling here in this middle bit, and over the next hour, you can see the swelling had started to spread out. So technically, according to our guidance, docetaxel doesn't require you to cold the plastics, but because it was an exfoliant, we did cold the plastics and they did come down and do a washout. And you can see here 3 weeks later, this is the extent of the redness. The gentleman had the washout. This was over a Christmas period. This happened about 2 days before Christmas. He had the washout. He did go back to the John Radcliffe to be reviewed by the plastics before Christmas. We arranged with our triage team that they would ring him and assess him daily over the Christmas period. He was given a course of antibiotics, and he did go on to make a full recovery. You can see his skin is looking quite dry there, and he was recommended to keep applying lots of moisturizer and to massage that area and to go with deeper massage as soon as the redness the redness cleared more. And he did continue with his docetaxel in his other arm. So the aftermath of the crisis is think about the patient and their family, think about the nurse who cannulated the patient, the doctor treating, because quite often the doctors who will review our extravasation injuries, we may be more experienced than they are, and everybody will be looking at the policy, think about the wider team, think about what went well, what could we have done better, what learning is there, and how to prevent and go forward. And in short, that is that. Thank you. Have you got any questions? I still see a lot of people here, but I'm not sure if yous are on mute.
Hi, Liz.
Chandra, hiya, Chandra.
Hello, hello. I just wanted to know, have any nurses been affected by extravasations? Have they, got it on their hands or their skin in any way? Has it ever affected any of them?
No, it's always been that it's been in the local tissues that it's the extravasation. I think which we will probably cover, I'm hoping, in another session when we do a revamp on spillage, which will be slightly different. So we'll cover that then, Chandra.
Okay, thank you.
All right. Any other questions? Okay, I've ran over time and I've seen quite a few thank yous, so I'm going to stop there. Thank you all, everybody. We'll see you on the 27th, Thursday, the 27th of April. Thank you.